So we were enumerating the steps involved in genetic engineering. So first of all, that we saw that you require insert DNA or gene of interest to be isolated. Then vector DNA is required to be selected. That it may be a plasmid DNA or fast DNA, and many other different types of vectors are there. Any one of them might be required depending on the purpose. Enzymes of different types are required. Most important among them are restriction endonuclease, ligases, and different types of modifying enzymes. The next thing that will be required is a host cell within which you have to transfer the DNA. Basically, it is E. coli DNA at the initial stage, in which the particular, if it is a E. coli cell, particular recombinant DNA that is being made is transferred to this E. coli cell just for the sake of storing, and then it is kept in either in liquid nitrogen or minus 70 degrees centigrade deep freezer for future use. And another thing that you require is electrophoresis instruments, just for the sake of monitoring the changes that you are making, just to monitor that the recombinant DNA has been synthesized, it has been transferred to a host cell, and that is the recombinant DNA. You can take out this DNA and you can check by making the same restriction digestion, and then you can run on a gel to see whether the same band is being formed or not. So to verify or to monitor all these changes that you are going to make during the time of recombinant DNA technology, you need electrophoresis instrument also. So we will now see in our next class different types of enzymes that are required. Let me give you a brief idea about that. So enzymes that are required during genetic engineering, you may classify them into two types. One is called modifying enzymes, another is restriction endonuclease. So restriction endonucleases are type of enzyme, and these are basically endonuclease, that is they introduce cuts inside the DNA, but the name restriction is there. That means these DNA are provided, these enzymes have the capability to restrict the entry of foreign DNA. Restriction endonucleases are usually present in many bacteria just to protect their cell from foreign invaders like bacteriophages. A bacteriophage, when it delivers its DNA inside a E. coli or any other bacterial cell, it might replicate inside it, or it might be degraded or aborted. Most of the cases it is aborted because the bacterium, it possesses a sort of enzyme, those are called restriction endonucleases, that cleave this DNA and do not allow them to replicate inside. But many a time the phage DNA is successful in replicating inside by a mechanism called modification. So all this restriction endonuclease system has two components. One is restriction, another is modification. The modification is methylation. So this system is together called restriction modification system. A restriction modification system is present in a bacterial cell, that with that system it either allow the DNA to replicate inside, the foreign DNA to replicate inside and complete its life cycle, and when it does, then modification phenomena function is being taking place. And if it does not allow the foreign DNA to replicate, then the restriction part of it is active. So both the system are together present, that is called restriction modification system. It might allow the cell, the phage DNA to replicate, or it might restrict also at the same time.